I'm Ananya. I'm a junior at Brookline High, and I researched a cancer gene called CDKN2A with Dr. Sumeda Dahal. So my main goal, so going into this program, I wanted to learn more about lung cancer. Um, and I focused on two subtypes of lung cancer after doing some basic research on them, which were lung adenocarcinoma and lung squamous cell carcinoma. So a little bit of background on the cancers. So lung adenocarcinoma, this is found in the peripheral regions of the lung, and this is usually diagnosed in smokers and non-smokers, but actually more commonly in non-smokers, which I was kind of um, I was surprised by that. While lung squamous cell carcinoma, this comes from the squamous cells in the central bronchi, and this is usually diagnosed with patients that have a smoking history, and these are primarily males that are diagnosed. So a little bit of background on the gene that I researched. It's called, the full name is cyclin-dependent kinase inhibitor 2A. So this gene is located in chromosome 9p21, and through its process, it produces two different types of protein, which are P16 and P14, and it produces these proteins through a process called alternative splicing. So P16 protein, this protein kind of acts like a brake, so it stops proteins from advancing into something called the S phase, which is basically the phase where DNA is copied. And by blocking these proteins, P16 is able to stop the cells from dividing further. Um, and P14, this also stops damaged cells from, or this stops damaged cells from dividing. And there's another protein that's involved in this process called MDM2. But this protein tries to get rid of another protein called P53 protein, which is necessary to stop cancerous growth from developing. And basically, the function of P14 is that it stops the MDM2 protein from blocking the P53 protein. And the process of alternative splicing, this basically just allows a gene to um, code for different types of proteins that have different functions. So this gene is also a tumor suppressor, which helps regulate the cell cycle. And some of the side effects of this gene are pancreatic cancer can develop and melanoma. So going into my data, this is the box plot that I had for the two cancers. And as you can see, there's a really high expression of this gene in the lung squamous cell carcinoma, and versus the group of patients that didn't have this disease, there's a much higher expression of it versus this one. And there's also an overexpression of the gene in the lung adenocarcinoma. And then for the survival plot, uh, this basically shows, so the red is the higher expression of CDKN2A versus the lower expression. So there's a longer survival for patients that have higher expression of this, which kind of makes sense with the fact that this gene is a tumor suppressor, meaning it's trying to stop the cancerous growth. So that kind of and that made sense with the data I was given. And then going into the mutations that develop for the different lung cancers. So this gene, it has um these are these are the graphs for multiple different types of the cancers. So there's for the higher expression of the alterations that happened was 55%. And the most common mutations that occurred were truncating and missense mutation. So missense mutation is basically a genetic mutation where there's a single change in the nucleotide base. And the truncating mutation is a type of mutation that causes the production of a shortened protein. And for my survival plot, this red line, this kind of shows the the probability of overall survival for a group that has that like there was a mutation that occurred versus the blue, which is like the unaltered group. And I thought this was pretty interesting because there's actually a longer survival for the altered group, which I didn't really understand because if there's a mutation, like the cancer would probably get worse, but there's a longer survival. And um, from this, like I tried to like research why this happened, but I wasn't really able to get like a super clear answer. So I just made like a prediction that like the tumors with the alterations might like respond better to certain types of treatments, or there's like a really slow growing tumor. Um, and then for the KM plots, the survival KM plots that I researched with this gene, this basically the overall survival just shows um like the probability of a patient's survival over the course of years. So there's a pretty like steady decline, which shows like patients live for around like 14-ish years. It's like 170 months. Um, and for the disease-free survival, this basically just shows like how long patients can stay cancer-free after a certain treatment. So this is a less extreme decline as the overall survival. Um, yeah. And going into like some extra data that I looked into, I can't really zoom in anymore, but oh wait, here. So I like along with CDKN2A, like with Sumeda, we also explored like a couple other genes that were a mixture of oncogenes and tumor suppressors. So there was I researched TP53, which is a tumor suppressor, EGFR, which is an oncogene, RB1, I'm pretty sure is a oncogene as well. And then ALK, it's either a tumor suppressor or an oncogene. I don't exactly remember. But this is basically a heat map. And I just wanted to like compare CDKN2A versus the other genes that I researched. So this heat map basically just shows that there's like a higher alteration of CDKN2A and like lung squamous cell carcinoma, and there's a pretty like steady or like equal percent of alteration in the other genes, while ALK, this has like no expression at all in um either of these cancers. And basically, in conclusion, I was able to learn a lot about like CDKN2A's role as a tumor suppressor, which kind of showed like this plot showed how the mutations can kind of show like unexpected survival outcomes because like I found out that the altered group survival is longer, which I didn't expect. And I was able to research a lot into like different types of genes, like tumor suppressors and oncogenes. Um, and yeah. Oh, and I also use like these are the references that I use, which are GPA, CBioportal, and I like learned more about CDKN2I in the Natural Library of Medicine.
Great job, Ananya. You have a full-fledged poster.
Yeah.
So was it the first time you ever made a scientific poster, or have you done it before?
I've done like over the summer, I did a summer camp just like in Boston, and we made like a scientific poster as well, but it wasn't like related, it wasn't cancer research. So this was
Wow,
really this was a really exciting opportunity for me.
great.